Good morning. I'll be presenting Nano-Therapeutics for liver disease, a site-specific drug and a small molecule delivery. So last Monday we heard a lot about the nano delivery system, the translational value, and the most recent advanced development as a Nano-Therapeutics. So I'll just try to cover the nanotherapeutic definition, classification, and characteristics, targeting strategies which are available. So liver-specific delivery system, I'll be especially focused about the liver fibrosis and some studies that we are doing on the basis of this nano delivery system. So nanotherapeutics are the therapeutics drugs, peptide, protein or nucleic acids loaded with a carrier in nanometer range. And they have the molecule which cannot achieve an effective and targeted delivery due to the biological, biophysical and biomedical barriers can be engineered as a nanoparticles. So most the advantages of this nanoparticle delivery system are there is especially nucleic acid and short life drug that can be administered to the in vivo system. The molecule which have less viability in the in life system which can use as a pharmacological to enhance the effectiveness of this molecule. Also, we can use these nanoparticles to reduce the toxicity and other side effect of this molecule. Reduction of the drug blood levels fluctuations can be addressed with the nanotherapeutic systems. Broad spectrum of the administration route can be enhanced from the one to another route. Controlled drug release can be also managed with the nanoparticle system. And of course, the active targeting to obtain a site-specific delivery can be achieved only by the nanodelivery system. So uptake and distribution of the nanoparticles depend on their size. So more than 400 nanometer size is quickly captured by the reticuloendothelial systems. So the people use below the 400 nanometer size, and less than 200 nanometers molecule prolonged blood circulation and with a low rate of reus uptake. So any molecule which ranges from 1 to 100 nanometers can be used as a nanoparticles. So they are polymeric, inorganic and lipid based. Polymeric are the precise molecule control the particle characteristics. They are flexibility in payload. We can load in the higher size of the molecule using this polymeric, but there are certain limitations, possibility of the aggregation and toxicity because of the size. Inorganic molecule we use most for the diagnostic and therapeutic use, and less because they are also having a size issue, and the druggable target where we can use is also limited in the inorganic. But for the lipid-based liposomes, lipid nanoparticles and emulsifications were used for the formulation simplicity, the range of physiochemical properties which can enhance. High bioavailability can achieve by the nanomicelles-based systems. Payload flexibility is also find very good in the lipid systems, and low encapsulation efficiency. So lipid-based nanoparticles are most common class of FDA-approved nanomedicines. So we are using the lipids in the for the as a nanotherapeutics. So size and shape diversity of nanoparticle based also is characterized very well. So macrosphere is like 100 micrometers, and this is 1,000 times lower than the microspheres. And the range between the 1 nanometers to 1 micrometers, the all biological system is from atom to the animal cells. So what we are using mostly up to 9 nanometer to 100 nanometer size, which can easily deliver to the even animal cell or human cells. So nanobiomaterials application as a drug delivery system has been rapidly evolved through the multidisciplinary field of nanotechnology due to its predominant opportunities related to the therapy of life-threatening disease. So before entering into the body or life system, there are certain biological barriers which allowing this molecule to a tissue-specific drug delivery. So once they get invested in the body, the first barrier is intravascular barrier. So where this molecule should be safe with a phagocytic system, they should not be uptaken by the phagocytic activities. They cannot destroy. So the molecule should be characterized well to cross the intravascular barrier, and safety should be there in the phagocytic system. Once they reach to the blood compartment, they have another system of endothelial barrier. So endothelial permeability should allow this molecule to reach the site of the cells where you want to deliver. So second barrier, the molecule cross the endothelial barrier to reach the particular cell type. After crossing the endothelial cell lines, they have to travel the interstitial space to reach the particular cell where we want to target. So there is a two different part that we have. They limit their efficacy or reaching capability of this molecule. There's a high interstitial fluid pressure, and second is the dense ECM. So dense ECM may not allow this molecule to reach the particular size. So molecule should be characterized to considering all those points, what kind of ECM we have and what kind of interstitial pressure we have. Once they reach to the cells, again we have to see how this molecule will be internalized by this cell system. So if they have a receptor, they can internalize to the receptor mediated, otherwise they can be the liposomal activity, phagocytic activity, they can internalize from the cell surface. Once they reach inside the cell, then they can reach to the cytoplasm or even a nucleus. So what are the targeting strategies that are available? So any nanoparticle molecule we can characterize on the basis of surface and material properties. We can modify the surface. This nanoparticle could be self-assembled peptide. Peptide can aggregate and they form the nanocarriers. Considering the immunogenicity, we can add with the pegylation. And cell coating also can be modified at density of toughness. So there's sustained release and easy to release the molecule that can be also be modified on the surface modification. The architecture of this molecule also playing a major role, where the small size can reach to the particular cells. The big size can be stuck in the reticular endothelial cell, cannot be targeted to the particular cell type. Shape is also very much important, where they can be entrapped by the neutrophils and other immune cells. Charge is also playing very important role, that is anionic or cationic. So on the basis of this, we can deliver this molecule. Talking about the targeting could be achieved by the receptor mediated. So sending any nanomolecules in the liver is not that much difficult, because anything you send that can reach to the liver. But sending it to the particular cell type can be achieved by the target affinity. So how we use this? So how we can achieve this, whether on the hepatocyte or another, they have used the mannose or dextran mediated, so they can be easily uptaken by the hepatocyte. Antibody ligand binding can be utilized to target any nanoparticle to the cell type. Surface receptor ligands can be attached to the nanoparticles. They can direct it to the particular cell receptor mediated. And vitamin A also use, vitamin D also use, vitamin A also used for some certain cell specific receptor. Most advanced people are using the aptamer peptides. They can be recognized by the multiple cell types. My talk is focused mainly about the fibrotic. So we are considering to deliver nanoparticles to activated hepatic stellate cells. So what are the strategy we can use to deliver any molecule to the activated hepatic stellate cells? That is based on RGD collagen type receptor, because we know the HSCs produce lots of collagen, so we can utilize this our cell to send the hepatic stellate cell. So we tag these nanoparticles to the RGD, even the PTGBT receptor, which is especially present when the cell became activated or profibrogenic. Mannose-6 phosphate delivery system and insulin growth factor 2 also utilized to send in the liver, where they can cross the matrix and they can reach to the hepatic stellate cell. Adenoviral transfection mediated ligand with the PTGBT is also can be utilized to send the hepatic stellate cell. Antibody, especially the cytosine C1-3CAB, used for the sending this molecule to the hepatic stellate cell. Vitamin A containing liposomes. So vitamin A is dicey because of the most of the quiescent HSCs, they have the vitamin A storing capacity. So this molecule can bind to the quiescent HSCs, and they can less bind to the activated HSC, because by activated HSC they lose the vitamin A. Other cell type in the sinusoids we also can target for the fibrosis, that's the Kupffer cell and liver sinusoid endothelial cell. So certain markers like CD206, Foucault, CD36 and CD68 are expressed on the Kupffer cells. So we can utilize this receptor to send this molecule to Kupffer cells. Nanoparticles like chloridinate liposomes can be easily uptaken by the Kupffer cells. So this molecule, if you want to target the Kupffer cells for the fibrosis, we can utilize this. For endothelial cell, we have lipoxygenase and SRC. LSEC scavenger receptor, which is present especially on the LSEC, so we can utilize that to send this. Even hyaluron receptor and CD44 were widely expressed on endothelial cell during fibrosis, so this strategy also we use. So liver-specific nanotherapeutic targets, whether it can be any cell types, we can utilize the galactose, EpCAM for the HCC, integrin for cholangiocyte and activated hepatic stellate cells. PTGBT is again utilized for hepatic stellate cell. Hyaluron for sinusoid endothelial cell. Mannose is for Kupffer cells. in hepatic islet cell, how we can send to the any molecule which can be taken up by the hepatic islet cell. So in response to any fibrotic stimuli, the receptor is overexpressed on the surface of the hepatic islet cells. So our nanomolecule can be tagged with a ligand which can go and bind on the receptor, and when they bind and hold the receptor, they can release the nanomolecule in the periphery of the hepatic cells on the zone. This molecule can be self-internalized or can be internalized to the receptor. Once they reach to the inside the cytoplasm, they can be proteasomal degradation, liposomal degradation, and they can release this molecule, and they can further release the drug inside the cells. So these are some examples where people have used some different strategy to send the nanomycels or nanoparticle for the fibrosis. Most of them have used to target the hepatic islet cells because it's the most abundant cell which is present in the fibrosis. So on the basis of these strategies and utilizing this knowledge, we are performing some studies at our center. So I have a few studies and data I'm going to show. So fenretinide as orphan nuclear receptor, which modulate and emulate the portal pressure, reduce fibrosis preclinically. So in this study, what we have done, we have identified the orphan nuclear receptor pathway, which is a immediate early response genes, and they involve in a diversity of cellular process. And the orphan nuclear receptors regulate TGF growth factor negatively. So deficiency of NR4A1 also can exacerbate the TGF beta associated liver fibrosis. So what is the normal condition? NR4A1 is present as a protective receptor in the healthy. But in fibrosis or persistent activation of TGF beta, they down-regulate the NR4A1 expression. Once the NR4A1 is present in normal, they inhibit inflammatory and profibrotic genes. So during fibrosis, NR4A1 levels goes down, TGF beta goes up, and TGF beta promote the fibrosis to the activation of hepatic islet cell. So what we thought, can we target this NR4A1 to modulate or enhance its expression to overcome with the attenuated fibrosis and its complications? So the first challenge was how we can achieve the molecule that can regulate the NR4A1 pathway, because that was orphan nuclear receptor. So first in silico drug library we identified the molecule that called fenretinide that has a affinity structural activity relationship with the NR4A1 receptor. So this fenretinide molecule was the trans-retinoic acid derivative we utilize to target this receptor which is present on the HSCs. And this fenretinide is a potential, the characteristics or they have anti-neoplastic and chemoproteinic activities. They are orally active but poor aqueous solubility, and consequently they have low bioavailability. In clinical studies, they have evaluated fenretinide, have shown dose-dependent systemic toxicity and poor tolerability. So here where the challenge was how we can send this molecule, or we modify this molecule using some delivery system. So we have generated, in collaboration with a group from Italy, we have generated this molecule fenretinide with a nano formulation. So the main challenge was the pure fenretinide was a very low solubility. So due to this reason, they have to send a high concentration to get an effective effect. So in nanofenretinide, what we have used, we got the solubility is 1,000 folds higher using the nanocapsulation. So what we have done in nanocapsulation, we have taken the skeleton of fenretinide, core molecule, and 2-hydroxypyrrolene beta-cyclodextrin formulation. We entrap this fenretinide into that, and then we characterize what is the physical property of this molecule. So when they have checked fenretinide solubility was less, after adding this nano formulation, the peak is shift toward the left, and we got there is a solubility is high. And this was further confirmed with the in vivo plasma pharmacokinetics levels of the nanofenretinide. So after single administration of the 5 mg per kg, the Cmax was achieved. Cmax was achieved at this NCI is the National Cancer Institute who provided this capsule for the clinical use. So they have limited Cmax at higher dose, while in nanoformulation capsulation after that we got a double of the concentration at the same dose. Even when we have administered intravenous, we found the nanofenretinide has just double of the concentration in compared to the pure fen, which is even cannot use for the clinical purpose. So in comparison to NCI formulated nanofenretinide, when we utilize this skeleton and we encapsulated with nanofenretinide, we found a very good solubility and the concentration of this molecule in vivo. Not only the concentration, the release of this molecule was also addressed by using this absorbing phase, which is the biphasic and is the lipid carrier. So when they have a very good hydrophobic hydrophobic environment, then they release the drug. So hydrophobic environment is created with the nanomicelles because this is lipid based, and when they bind with the cells, they release this molecule in good concentration, and sustained release can be achieved up to 24 hours. So this was the characteristics of this nano formulation that what we use. The polydispersity was 0.2 and size was 300 nanometer after including the molecule. The drug loading capacity was significantly high. It was 8.2%, which is sufficient to send any drug. The phenretinide solubilization was achieved in dose-dependent concentration. This was also very good. The most important thing, the release of this molecule at the pH dependent, because this molecule can be given orally and intravenous and system. So we wanted to target the give drug orally. So in low pH, this molecule is not releasing the phenretinide up to 72 hours. But when they reach to the intestine or the normal pH, this molecule is released in a good amount. So they are safe with orally also. In toxic effect, this nanophenretinide we did not observe any significant change in the body mass or any change in the hepatic or in the hematological parameters. So how we use this molecule? We first investigated this molecule in the primary hepatic stellate cell system. So we isolate the primary hepatic stellate cells and we allow them to activate in vitro, and then we treated with the nanophenretinide. And we observe this nanophenretinide, as we expected, they are reducing the hepatic stellate cell activation. In the gray and black bar, you can see during the in vitro activation, we found all the activation marker was significantly high at different passages. While when we treated these cells with the nano formulation, we found a significant reduction in all the hepatic stellate cell activation marker. Also, they are promoting the apoptosis of the activated hepatic stellate cell. After the in vitro experiments, we have utilized the animal model of liver cirrhosis, where we have administered this drug 4 times a week for 2 weeks in the macronodular cirrhotic animal models, and we performed the hemodynamic analysis and fibrosis and other molecular, and we found due to reduction in the hepatic cell activation in vitro, we found there's a significant reduction in the portal pressure and intrahepatic resistance. So in the phenretinide treated animal, we found up to 38% reduction in the portal pressure and 40% reduction in the intrahepatic resistance. The portal blood flow was also reduced in the treated animals, and no significant change we observed in the mean arterial pressure. And in vivo also we have checked. After giving the phenretinide, we found the NR4A1 gene expression was significantly high. So increased NR4A1 gene expression was correlated with the alpha SMA, downregulation, MMP2s, and reduction in the fibrosis, even the TJ-beta levels. In tissue histology is also confirming the reduction in the fibrosis in the phenretinide-treated groups. So overall, what we got, phenretinide enhancing the NR4A1 expression, inhibiting the hepatic stellate cell activation, and reducing the fibrosis and portal hypertension. So in short, what we have done, we first characterized this pathway in the diseased human, and we validated in the animal model. Later, we identified this molecule in silico, and we identified a target which can modulate this pathway. So we also addressed the limitation of this molecule using the nano delivery system, and then we utilized this nano delivery system in vitro and in vivo, and we observed there was a significant reduction in hepatic cell activation, fibrosis, and portal hypertension. And this molecule we have submitted for international patent. So overall, modulation of NR4A1 activity by phenretinide is a promising avenue to treat chronic liver disease and portal hypertension. In study 2, what we have done, we try to check the efficacy of this molecule, whether this is 100% going with the NR4A1 or they have a pleiotropic effect on other pathway. So there is 2 strategies, either we can use the knockout animal, or we can knock down the particular gene in the cirrhotic animal. So what we have done, we have developed a cyclic RGT conjugated dipeptide nanoparticle based injectable, sustained and targeted delivery system of siRNA in experimental liver cirrhosis. This work was done in collaboration with ICGB, and they have a peptide chemistry lab. So challenge of the gene delivery is to design a safe and efficient delivery system, protection from environmental condition, basically in mechanism of the fibrosis, enhance cellular internalization and control release. So people have explored the HSA coupled mannose-6-phosphate to deliver retinol modified liposomes and cyclic peptide. So limitation was the lack of specificity, target both quiescent and activated hepatic stellate cell, affect the scavenger receptor on endothelial cells. So we have to be very careful to deliver this molecule, whether they can go and specifically knock down the particular gene in the hepatic stellate cells. So what skeleton we have used, that is arginine alpha beta dehydrophenylalanine, that is RUD-alpha. It is a 20 nanometer long peptide. And this peptide we have used, already published this peptide, they have used for the cancer and translational and the preclinical studies. So this peptide they have modified, and we wanted to send to the HSCs. So what we have used, we have added the RGD, this is arginine aspartate acid residue, which can bind to the collagen 1 receptor. So after interacting with the RUD-alpha, that is the peptide and RGD target, we characterize this whole nano-delivery system, and we found this nano-delivery system has a good zeta potential levels. We can utilize the size and dispersion using the daylight scattering. So surface is like minus 9.57 millivolt. The hydrodynamic diameter of the RGD peptides was 2 mg per rebel. It was less than 200 nanometer after incorporating with the RGD peptide. TEM analysis also saying lyophilized suspended RGD have a 16450 nanometers. So this molecule we can utilize considering all the physicochemical property of this RGD system. So first we checked the in vitro biocompatibility. So whole RGD peptide without any siRNA we have checked in 4 cell lines, and we found there's a very high concentration of the siRNA or RGD we got, only RGD we got the cytotoxicity. 773 micromolar is a very high concentration in considering the can create the cytotoxicity. In hemolysis also we found even high concentration. They are not producing any hemolysis effect in the system. After incorporation with the siRNA also we got the similar results. Up to 200 up to 200 micromolar concentration, there is no toxicity was observed. Even hemolysis was also addressed properly. There was no hemolysis effect after the incorporation. Immunogenicity is another challenge. Peptide-induced immunogenicity, we have checked the interferon gamma response in the T cell with D cell, DCs and T cells, where we have used the positive control meso-1 image. There is immunogenic peptides. In corresponding to the immunogenic peptide, this RGD peptide is non-immunogenic. They are not having producing the sufficient interferon gamma concentration. After the immunity, we have to check whether this molecule can be uptaken by the hepatic stellate cell or not. So what we have done, we have cultured the hepatic stellate cells, and we have not treated with the this fluorescence tagged nanopeptides. So we observed no fluorescence. While when we have this RGD peptide fluorescence tagged, we incubated with the hepatic stellate cell, so they have gone to green fluorescence, which is the 100% or up to 80% nanofluorescence was uptaken by the hepatic stellate cell, which was confirmed by the confocal images, and we quantified further with the FACS analysis. So we have addressed how this RUD-alpha RGD peptide self-assemble, and we have checked there's a physical property as in we characterize the nanoparticles. We have checked the in vitro toxicity and in vivo toxicity in biodistribution. We have also checked whether this is going to the sinusoid in comparison to other organs. So they are reaching to the sinusoid of the cirrhotic animals compared to the control. And also we have confirmed whether this siRNA is able to reduce the gene expression as well as the protein expression. So after this, what we got in final, the nanoparticles, RGD peptides, are the self-assembled, can be given to the animal. They are safe. In vitro toxicity was also addressed. Once they reach to the sinusoids, they are picked up by the hepatic stellate cells, and they are down-regulating the NR4A1 gene, and deficiency of NR4A1 gene is confirming there's a fibrosis progression. So in the previous study, we have shown already that fenretinide can utilize to overexpress the NR4A1. So here we are going to now check whether this fenretinide can be effective through the NR4A1 pathway or not. So RGT peptide delivery system, what we developed for a small molecule, can be further evaluated and validated for therapeutic purpose in various pathological conditions. So this was the hepatic target. We have utilized the non-hepatic target also. So where we utilized the vascular endothelial growth factor, mediated restoration of mesenteric lymphatics, vascular function, and experimental cirrhosis. So gut lymphatic vessels and drains, they are drainage pipes. So gut lymphatic system comprise the lacteals collecting lymphatics and mesenteric lymph node and lined by the lymphatic endothelial cells. There are certain surrogate markers, the protoplanin, VEGFR3, and LYV1. So in physiological condition, they're maintaining the body fluid levels, absorb the transport fat.
Dinesh, would you wrap up a little faster? Here.
Yeah, okay.
Yeah.
Sure. So I can give the final focus on this nano-engineered. So we have to target the lymphatic endothelial cells. So we utilize the nano-engineered molecule where we can the human recombinant protein VGFC using this nano capsule. So in the nano capsule, what we have designed, we have taken the aqueous phase molecule where the molecule inside this aqueous phase molecule our recombinant protein is safe, and this was shielded with the organic phase for sustained release and stability. And this whole nano capsule was tagged with the VEGFR3 receptor, so this can go and bind to the cell which have a VEGFR3 receptor. This acts as a ligand. So the specific delivery and targeted to VEGFR3 receptors, this can be achieved using this strategy. So this was the physical characteristics of the molecule, size and dispersity and release of the molecule that we have checked. We have checked the biodistribution of this molecule in the cirrhotic animals in compared to the control, and we found that most of them are expressed in the mesenteric, kidney, and the duodenum. So this was what we were trying to give, not to send into liver. So we found very less concentration in compared to the other organs. That was majorly present on the mesenteric, kidney, and the duodenum. Later, we have used these mesenteric lymphatic systems to check whether this molecule is going to bind with the cells which have a LYC1 receptor, and the concentration was high, and the specification with this VEGFR3 was high in CCL4 cirrhotic animals. So in this same condition, when we given the engineered VEGFC, we found the VEGFC is high in the protoplanin positive cells, and where we have found not only the mesenteric, even the lymph node. So after giving the VEGFC, we found in the cirrhotic animals, the protoplanin levels are positive. So the idea was giving VEGFC was to get the pro-lymphogenesis. So this can be addressed by the expression of VEGFC, protoplanin positive cells. So we found after treatment with the VEGFC, we found the protoplanin cells was positive, and the diameter was also reduced. The dilated vessels were less, and HSC score also we got the most of the cells VEGFC were positive in the after the treatment with VEGFC. The protoplanin cells were positive in the VEGFC treated groups. In the animal, we have done the macrocity. We got there's a reduction in the ascites when we have treated these animal with VEGFC. So finally, what we have checked in the cirrhotic animal and human, we found that there is a lymphatic system is dysfunctional, and their VEGFR3 cells were expressed. And we utilized the ligand to send the VEGFC as a therapy, which targeted VEGFR3 positive, VEGFR3 receptor, act as a ligand. We given this molecule orally to the animal, and we observed the translational effect. We got the reduction in ascites, which have increased VEGFR3 frequency, and certain inflammation markers also reduced, and we also got the reduction in the perturbation in these animals. So I think I will skip this because this is another strategy where we use the silencer siRNA. So in final, the FDA approved nanotechnology product and clinical trials have immensely increased the, including synthetic polymer particles, lysosomes formulation, micellar nanoparticles, protein nanoparticles and nanocrystals, and many other in combination of drug or biologicals. Advances in nanomedicine, our ability to diagnose disease and even combining diagnosis with therapy has also become a reality. All key cells in fibrogenic process can be reached by the cell-specific drug carriers, and further research should address the key factors that determine the success of this nanotherapeutics. Thank you.